Hi guys, this is Jubilee, and we're going to be doing the liver, etc., and urinary systems TA review today. Just bear with me as we go through this. I'm going to try to explain this as much as possible to you guys, hopefully without making it too lengthy. Okay. But yeah, hopefully by the end of this, you'll be able to identify what all of these different pictures are of. And let's get started. So first we're going to go over the liver and all the gallbladder, pancreas, and salivary glands. So kind of the main functions of these are all accessory organs to the digestive system. And so they all kind of secrete things and make things that are going to help you digest food, right? And so since they are accessory organs to the digestive system, they're helping the digestive system do its three main roles or functions. And so we learned that those were the movement of food, the secretion of digestive juices, and the absorption of digested food, right? And one of Dr. Jonathan's big things is that one of the functions, or I guess one of the functions that the digestive system does not have is it does not digest food, right? It secretes digestive enzymes that will help you digest your food, and it absorbs that digestive food, but the digestive system in itself does not digest food. It's the enzymes that it makes. And so these accessory organs are going to help do that as well, right? So the salivary glands are going to help the esophagus of the digestive system to move food by lubricating that food down the esophagus so it travels more easily, right? The salivary glands, the pancreas, and the liver all secrete digestive enzymes or juices that are going to help with that digestion of food. And then also to help absorb the digestive food, the water, and the electrolytes. That's a lot of that is going to have to go to the liver. And that's why the liver is so important to the digestive system. We're going to learn in a second that it is the first organ that receives all of the blood that goes through your digestive system. So it is the first organ that's going to receive all of the nutrients, all of the toxins, or all of the drugs or anything that your digestive system has absorbed. Your liver is going to be the first thing to process that. So that's really important as well. Okay, so we're going to talk about the liver, and the liver has many functions. It's a really important organ. And again, that's why it is the first organ that gets all of the blood from your digestive system, right? So food's going into your digestive system, blood's going into your digestive system, and that food inside the tubes of your digestive system is getting absorbed into that blood, right? And so all this blood is absorbing all of this food that your intestines have just absorbed, and that's all going to go straight up to the liver, and the liver is going to be the first organ to get all of that. And so it's going to decide how to separate up all of those pieces of food, all the protein, fat, everything like that, and divvy it up to the rest of the body. Okay, so it has a lot of functions in doing that. So it's going to help maintain your blood glucose levels. That's so that if it's been a while since you've eaten, it's going to help you maintain blood glucose levels. And if you've just eaten, it's going to make sure that you don't just dump all of that glucose into the blood straight away, right? So it's going to help divvy that up so you're not always either just starving or completely saturated with glucose, which is going to cause problems. It's important for lipid metabolism and transport. So your liver is going to have a say in that. Plasma protein secretion. And so basically it's just what's going to decide how much glucose goes where, how much fat goes where, and how much protein goes where. So it has a really big role in metabolism, okay, in divvying things up and giving it to other organs. It's the kind of like the distributor of glucose, fat, and protein. Okay, another thing it does is it secretes bile. And so bile is going to be stored in the gallbladder, and that's also going to help in the small intestines to break down food. Everything that's in the bile is going to help with that. And then in the liver, it's also going to break down drugs, right? So if you eat something bad, if you eat something toxic, or if you eat a medicine that's prescribed to you, that your doctor has prescribed to you, your liver is going to be the first thing that's going to start breaking that down immediately, right? So if you drink too much, if you have a bunch of alcohol, that's why people say you're killing your liver, right? Because your liver is going to be the first thing to get it. It's going to be the first thing to process it. And that's kind of like the first protective barrier against any toxins, against any drugs, and stuff like that. So the liver, the main cells in the liver are hepatocytes, right? So we're going to talk a little bit more about that in a second, but hepatocytes. And so these main cells in the liver has exocrine and endocrine secretions. Okay, so whenever we say exocrine, that means it's something that's being excreted to the outside, right? Exo. And whenever we say endocrine, that's something that's going to be excreted towards the inside. And so while we might not think about bile being an exocrine secretion, it really is being secreted outside of your body, because it's being secreted outside of the cells in your body, and it's going into your digestive system, right? Everything that's in your digestive system is not a part of you, it's just the food you're eating. And so you're excreting that bile outside of the liver, outside of the hepatocytes, into the lumen of your digestive system, right? Whereas the endocrine secretions of those hepatocytes are going to be things like glucose and plasma proteins, because those are going to be things that end up in the blood. Okay, so those are going to be things that end up in the blood, and that's staying within you, right? Your other cells are going to absorb that, and that's going to be within you. So that's going to be an endocrine secretion, right? And so we don't want to mix up these exocrine secretions and these endocrine secretions, right? We don't want to be putting glucose and plasma proteins, these big nutrients, back into the digestive system where we just got them from. But we also don't want to be putting bile, this really harsh chemical, back into our blood, right? Where it can go to the rest of the body. It's supposed to be digesting things, right? Not us. And so we need a unique way to separate these things. And so that's why the liver is set up the way it is. That's why it's kind of complicated, because it has a lot of functions, right? It has exocrine and endocrine secretions. And so we have these two different holes, basically, where things are going to be secreted and absorbed. So first we have bile that's secreted in the bile canaliculi. So you see these holes? It's like in between the sides of two hepatocytes. So this is a hepatocyte, this is one on the other side, and in between these there's this little hole, and that's where they're secreting all of this bile into these bile canaliculi. Okay, and that's separate from these big spaces right here. This is endothelium, so this is a big blood vessel right here. Okay, and so that's where we're going to be doing our endocrine secretions into the blood. So this is a big blood vessel, remember, and we said that in the liver we have sinusoidal capillaries with big gaping holes, right? So here are these big gaping holes so all this blood can go through, and that's where we have this thing called the space of Dease. The space of Dease is just a special name for the space in between the lining of the hepatocyte and the endothelium, right? The lining of the blood vessel. And so that's so because of this sinusoidal capillary with big gaping holes in it, blood can go through this hole and it can bathe the hepatocyte in blood, right? So we have all these microvilli to increase surface area, so all of this blood can come in contact with all of the surfaces of the hepatocyte, right? Because we have a lot that needs to be exchanged between this hepatocyte and this blood vessel, right? We've got all this nutrients and stuff coming from the digestive system that needs to be absorbed, and then this liver is going to process it, and it's going to excrete out its products back into the blood, right? So it can go to the rest of the body. So this liver needs a lot of time to process all of this blood and make those secretions. Okay, so here you can see kind of the setup of things. So this is an EM of it, right? And this is going to show you, so this is that little bile canaliculi. So that's going to be where our exocrine secretion is going to go, where the bile is going to go in between two hepatocytes, right? And then here, so this is a big continuous blood vessel, right? This is a big continuous blood vessel, and so this is our space of Dees, right, where there's the microvilli of the hepatocyte coming in contact with the big spaces in this blood vessel so it can touch all the blood, okay? And then these are the hepatocytes, right? And so here's kind of the diagrammatic view. So these are all our hepatocytes, and in between each hepatocyte in the middle are the bile ducts, the bile canaliculi turning into bile ducts, and that's being secreted outwards, right? And that's our little exocrine secretion, whereas the blood is coming inwards. It's bathing these hepatocytes. There's that space of Dees right there. It's bathing the hepatocytes so that on this side they can come in contact with the blood. On this side, they're coming in contact with the bile that they're secreting, right? So on this side, the blood is coming in and the bile is going out because the blood is coming in to come in contact with these, and the bile is being secreted by these cells. So here you can see this again. Let me take that away. So here you can see this again. We've got the bile canaliculi on the sides, and we've got this space of Dees on the other side of this big hepatocyte, this big cell inside the liver. Okay, so here we can see more pictures. So this is more zoomed out. And so the liver, because we have this difference between the exocrine and endocrine secretions, because we have the difference between these sinusoids with the space of Dees and the bile ducts, we have this special kind of way that the liver sets itself up. And these are called lobules. They're called classic lobules inside the liver. So it basically means like mini lobe, right? Mini lobes inside the liver. And so in these little lobules, you kind of have the same thing in every one. So in every lobule in the center, you have what's called a central vein. So these are all the central veins in there. And then on the periphery, on the outside of every lobule, you have these things called portal radicals, okay? Portal radicals. And we're going to have a zoom in on those later. But basically out here, you've got your portal vein, you've got your hepatic artery, and you've got your bile duct, right? And then you've got your central vein. And so we said, so this is the hepatic artery. So this is just bringing oxygenated blood to all the little hepatocytes, right? And then we've got the portal vein. The portal vein, remember, is that portal system. So it's bringing the blood that already went to the digestive system to the liver so that the liver can get impacted by the change of the digestive system, right? So the blood goes to the digestive system. It absorbs all the food that's being absorbed in your digestive system, and then it goes to the liver to drop it off, right? So this is a vein, but it's still coming to the liver, right? So we've got all this blood going to the liver, and it's all going this way towards the central vein where it's going to be drained out, right? So we've got blood going from the outside of the classic lobule to the inside, whereas all of these hepatocytes are creating and secreting bile. So bile is being secreted by them, and it's going outward to the bile duct on the outside of this classic lobule. So you see how we've got blood going inward towards the central vein, and we've got bile being produced and going outward towards the bile duct, right? So they're flowing in two different directions, and they're in two different spaces. So just make sure you keep that in mind. So here we can see that we've got our portal vein, artery, bile duct out there. And here's just a zoom in of those hepatocytes. So these are all hepatocytes in the liver. It's the main cell within the liver. And then you've got these big gaping holes. Those are those sinusoids, right? Those sinusoids, those big blood vessels where all the blood can go through and get in contact with the hepatocytes. Okay, so we talked about portal radicals really quickly. So this is just showing you a little more of what a portal radical is. So a portal radical is just a name for the whole unit on the outside of a classic lobule. So this is a classic lobule, and on the outside, the whole thing is called a portal radical. And it's made up of a hepatic artery, a portal vein, and a bile duct. So those are the three things that make a portal radical. Some of them have lymph vessels too, right? But they are always going to have those three things, right? Because we said the hepatic artery is bringing blood, oxygenated blood, so oxygen, to these hepatocytes. The portal vein is bringing the nutrients that got picked up in the digestive system to these hepatocytes. And the bile duct is taking the bile away from the hepatocytes because it's being produced by the hepatocytes, and it's taking it away off to the gallbladder. Okay, so here you can see a portal radicle, and so the big one that's not very circular is going to be the portal vein. Okay, so that's not going to have very much oxygen, but it's going to have loads of nutrients from the digestive system. These more circular ones are going to be the hepatic arteries, and so those aren't going to have nutrients from the digestive system, but they are going to have oxygen for those hepatocytes, right? And then this one, you see how this one doesn't have like thin endothelium, right? It doesn't have endothelium like a blood vessel. It has more of a thick lining to it, and that's because it's a bile duct, not a blood vessel. So that's our bile duct, and that's going to be carrying bile away from the liver towards the gallbladder. Okay, and so we talked about classic lobules. So here's our classic lobule in a diagram, right, with our central vein, and then we've got these little portal radicles on each side, right? And so that's one way to think of the way the liver orients itself. There's other ways to think about the way the liver orients itself. It's still the same liver and it's still the same setup. It's just different ways to name things. Okay, so don't let that trip you up. So we've been talking about the classic lobule, and that's just where the central vein is considered the center. So the central vein's in the center, and you've got your little portal radicles on the sides, right? But you can also think of it as a portal lobule, and so that's where the portal radicle is considered the center. Okay, and the reason to kind of use this is just you can kind of talk about the portal lobule as everything outside of the portal lobule is what this one portal radical is supplying. Right, so it's supplying oxygen to all of this and this and this out here, right? It's radiating out from the center of that portal radical through this portal lobule, right? And it's also where all of the bile is being picked up into this bile duct. So that's just a different way to think about it. Another way to think about it are Asuna Slayers. And so that's this one right here. And it's basically just, it's talking about these different zones, right? So you've got zone 1, zone 2, and zone 3. And it's basically just talking about the way in which blood is accessing these zones, right? So if we've got our portal radicals out here on the sides, on the periphery, blood is going to get to zone 1 way before it ever gets to zone 3, right? So if we don't have enough oxygen in our blood, the zone that's going to run out of oxygen first is going to be zone 3, right? Does that make sense? Because it is so far away from the bloodway out here. So if we don't have enough oxygen in this blood, zone 1 is going to steal it all up. Zone 2 is going to get some, and zone 3 isn't going to get any. So zone 3 is going to die off first if we don't have enough oxygen in our blood, right? Whereas in contrast, if we have a toxin in our blood, right? So if we have too much alcohol or we get poisoned or something terrible, we have a toxin in our blood, the first zone that's going to come in contact with that toxin is going to be zone 1. Right, so if we have a toxin in our blood, zone 1 is going to be the first zone to die off in our liver. Okay, so that's just a different way to think of it. Asuna Slayers, it's just talking about, which zone's going to die first, who's going to die first, basically, okay? So the cells within the liver, we have 3 main types of cells. So we've already been talking about the hepatocytes, right? That's the main functional cell in the liver. That's the one that does everything. So that's the one that's making all that bile. It's absorbing all those nutrients, and it's deciding who gets what and how to divide up the liver, the fat, the protein, the carbs, the fat, the protein, right? So those are the hepatocytes. So that's every single one of these. All of these in here are hepatocytes, okay? Hepatocytes. You've also got Kufur cells, and Kufur cell is basically just a specialized macrophage, right? So it is just the macrophage of the liver. So it's just this special name that we have for macrophages that are in the liver. So this is going to be a Kufur cell, right? There's a special stain that picks up these Kufur cells. So this is a Kufur cell right here, right? And then in this one, we don't have this special stain in there, but this is going to be our big Kufur cell right here. Okay, so you see how it looks different, like it's separate from everything else, but it's still nice and big, ready to eat something up. That's our Kufur cell, okay? And then we also have endothelial cells, right? Just like in any tissue, we have blood vessels in there, and so we have endothelial cells in there. And so like this big flat one right here lining this sinusoid, that's going to be our endothelial cell. These ones right here lining this tiny little capillary are our endothelial cells, okay? Those are all our little endothelial cells in there. So those are also, that's like the third type of cell that we're going to see in there. Here's an endothelial cell. Okay. And so in the bile ducts, there's four big things that you need to know are secreted into the bile ducts. So this is them: cholesterol, EGF, insulin, and IgA. Okay, so those things are really important with helping you with digestion, with helping, so insulin, right, is going to help tell the cells of your intestine, okay, we just got a big meal. They just secreted a lot of bile. There's a lot of insulin in here, so we need to start absorbing glucose, right? We need to start absorbing things. So that's what insulin's going to help with. IgA is a type of antibody, right? So whenever you're eating things and there's a bunch of, foreign objects in your intestine, there's going to be a lot of antigens, a lot of germs and stuff. And so we need to secrete IgA, an antibody, a special antibody that's going to help prevent you from getting infected by all of these random things that are in your GI tract, right? And then there's also bile salts and bilirubin, right? That's again what kind of makes up bile and is really important to bile. Okay, so here you can see all the little bile canaliculi in between the hepatocytes and how these bile canaliculi join together and eventually they make a bile duct, right? Eventually they're going to all join together and make a bile duct, and that's going to keep going and it's going to get into the gallbladder. So bile ducts from the liver are all going to empty into the gallbladder. And the gallbladder is really important for storage, right? Just like any bladder, right? Like the urinary bladder is important for storage, but unlike the urinary bladder, it is also there for concentration of the bile. So it's going to concentrate the bile, whereas in the bladder, it does not concentrate urine. So that's a big difference you guys need to know, right? The urinary bladder does not concentrate urine. And so this is the lining of the gallbladder. And so you can see we've got this simple columnar epithelium. That's how I know it's the gallbladder, right? So in this cross section that Dr. Johnson loves, it's like cutting it through right here. Okay, so we get two cross sections over the gallbladder. And so this one is a big vein, and you can tell because there's endothelium lining it. If we zoomed in, there would be endothelium. This I know is the gallbladder because it's got simple columnar, that thick epithelium lining it. This one I know is also the gallbladder because it also has that simple columnar. If you were to zoom in, you would see it. Okay, so here you can see this is the simple columnar epithelium of the gallbladder, right? We've got one layer of columnar cells, that's simple columnar. And again, that's going to help concentrate the bile in the gallbladder. So this is all gallbladder. This is where all the bile would be, and this is all liver tissue surrounding the gallbladder right there. You've also got plasma cells in the gallbladder, in the connective tissue around the gallbladder, and that's again just to help make antibodies that are going to go in there and help fight off germs. Okay, so just really quick about the salivary glands. So saliva is going to help prevent infections. It's really important in that. So it also, the salivary glands secrete IgA. Those are antibodies. So that's going to help fight off germs and stuff from getting inside. Lactoferrin, that's just, again, an antibacterial thing. So it's going to bind up the iron that bacteria need to survive. So it's going to help interfere with that, lactoferrin. And then it's got lysosome in it. And that's, again, going to kill bacteria. It's an enzyme that's going to kill bacteria. So it helps prevent infections, right? And it is constantly washing the mouth out, washing all that bacteria that's getting into your mouth down into the GI tract so that hopefully it can get digested and eaten up by stomach acids and stuff like that. So here are the salivary glands, and you can see we've got some serous glands and some mucus glands. The mucus glands are more bubbly and gross looking, I think. These are all serous glands right here. Okay, so in the salivary glands, an acinus is called a functional unit. So this whole thing right here, this whole unit of the salivary gland is an acinus. So this is an acinus. Up here would be an acinus. And you've got serous, mucus, and mixed salivary glands, right? So the serous ones are going to be here. The mucus ones are going to be here. And those are all going to collect into intercalated ducts and then into striated ducts. Okay, so these are going to be, this would be the acinus, this is the intercalated duct cross section, and these are the striated duct cross sections. And then there's also the pancreas, and so just like in the liver, the pancreas has endocrine and exocrine functions, right? So the exocrine secretions are going to be the ones that don't go into the bloodstream, that don't go to other cells within the organism, right? So those are going to be all of these cells out here. So these are the pancreatic Asuna cells, right? We learned that a little bit in the endocrine system. So these are all pancreatic Asuna cells out here, and they're secreting pancreatic enzymes, so digestive enzymes that are going to go into the small intestines, and they're going to help break down food. Okay, pancreatic enzymes. And then you've also got these islets of Langerhans, right, that we definitely talked about in endocrine. So these islets of Langerhans are where you're going to be secreting insulin and glucagon. And so that's obviously going to be an endocrine function because it's going to go into the blood. It's going to impact other cells. That's why it has an endocrine function. And so that's going to help not only tell the rest of the body, hey, we either need to absorb food or we need to save our energy for when there is food. But it's also going to tell these pancreatic Asuna cells, hey, we either need to, through insulin, we need to produce a lot of pancreatic enzymes, or through glucagon, hey, we need to stop producing pancreatic enzymes. Okay, so that's the islets of Langerhans and the pancreatic Asuna cells. Okay, so to the urinary system. This one always confuses people so much, and I know it's confusing. It's just because the kidney is just a ton of tubes, right? It's a ton of little tubes, and it's really hard to tell the difference between each one. So hopefully this will help a little bit. It's still really confusing, I know, but honestly, just do your best. Look at his labeled pictures and go from there. So the urinary system also has exocrine and endocrine functions. The exocrine function is kind of the one we're going to focus the most on. So the exocrine functions is basically the kidney is a giant filter, right? So, you're constantly producing waste, you're constantly drinking in water, and so the kidney is kind of what's going to filter out that waste, right? The urea, the uric acid, all of that. So it's going to filter out that waste, but it's also in charge of keeping the amount of fluid and volume of blood in your body constant, right? So I'm not constantly drinking water. I'm kind of like a camel, right? Where I'll drink a whole bunch at once and then I'll stop drinking until I'm hungry again or thirsty again, and then I'll drink a whole bunch of water, right? And your body cannot deal with all of those fluctuations in when you're drinking, when you're not drinking, or if you have massive hemorrhage, you bleed a lot or something, and your kidney needs to make up for that, right? Because your body can't deal with you not having enough blood in your body. And so your kidney is responsible for making those long-term changes so that over long periods of time, you do have a constant amount of blood, amount of extracellular fluid, and just volume of water in your body. And so your kidney really helps to do that. It senses how much water you have, and it can decide, okay, no, we really need to save water because we're dehydrated, or no, we need to get rid of water, get rid of waste because she's drinking plenty of water. And so we can just go ahead and get rid of that. And then we also have endocrine functions of the kidney. We're not going to focus as much on them, but the kidney does secrete erythropoietin, and that stimulates red blood cell production, right? So we said the kidney's in charge of deciding how much volume and blood you have in your body. So if it decides we don't have enough volume in here, we don't have enough stuff going through the filters of the kidney, we need to produce more red blood cells, it'll start secreting erythropoietin to tell you, hey, we don't have enough circulating through the kidneys right now, we need more, right? It can also produce renin. Renin causes aldosterone release, and that again just helps with blood pressure control, right? So if you have a giant drop in blood pressure, it'll start telling the rest of the body that it needs to compensate for these things. If you have really high blood pressure, stuff like that. So the kidney really helps regulate blood pressure. So talking about the kidney, so within the kidney, the nephron is the major functional unit of the kidney. So this whole thing right here, or the whole thing within the kidney, is a nephron. Okay, and basically the nephron is a bunch of leaky capillaries that create a filtrate. Okay, so in this glomerulus right here, there's a bunch of leaky capillaries, right? Because we said the kidney is basically a filter. And so as blood is going through the kidney, through these nephrons, there's a bunch of holes in here. It's fenestrated capillaries, right? We said there's holes that allow some things through, but not everything. So there are a bunch of leaks in here, and whatever leaks out, that's what forms the filtrate. Okay, so all the waste leaks out, a bunch of water leaks out, salt, potassium, stuff like that. But also a lot of things that we want to keep, like protein, stuff like that, leaks out. And that creates the filtrate. And so a lot of stuff leaks out of these capillaries, and a lot of it we want to keep. Okay, so it's kind of this non-selective leaky thing where everything kind of leaks out. Some of it stays in the blood, and then the stuff that we choose to keep, that the kidney wants to keep, gets reabsorbed back into the veins and back into the blood. Okay, and so in the kidney, in the nephron, it can control what to take back up, and what to excrete. And so whatever does get taken back up goes into the blood. Whatever does not get taken back up gets put out as urine. So that's the waste. Okay, so until it's finally excreted, it's called filtrate. But once we decide, okay, nope, we don't want any of that that's left, that's urine. And that's what's excreted. And so that depends on what the body needs, right? That depends on how hydrated we are, how much salt we ate in our meal, stuff like that, what we're going to reabsorb and what we want to keep back. So this whole thing is the nephron, okay? And so it's basically a series of many tubes, and that's why people hate the kidney, because it's just a bunch of tubes. They all do different things. They all look kind of the same, but also different. And so it confuses people, right? But this is a nephron, and so it starts out with this thing called the glomerulus, and that's where those leaky capillaries are. So we've got blood going in with those leaky capillaries, and so stuff leaks out and goes into the tubules of the kidney, and that's called the filtrate. Okay, so what it goes through first is the proximal tubule. The proximal tubule is going to reabsorb most of everything that was leaked out, and it's going to reabsorb it and put it back into the bloodstream. But the thing about the proximal tubule, proximal meaning it's more first, right? It comes first as opposed to the distal tubule. So proximal is first, it's primary. So the proximal tubule is going to reabsorb a lot of stuff, but it can't control what it reabsorbs, right? So it's going to reabsorb protein, it's going to reabsorb a ton of water and stuff like that. But it's not going to vary based on how dehydrated you are, right? It's just going to reabsorb this much water because it knows that it's supposed to, and it knows that too much was excreted in those leaky capillaries, right? But it doesn't care how dehydrated you are. It doesn't care how hydrated you are. It's just going to absorb what it's going to absorb. You can't control it. The loop of Henle is also uncontrolled. And the confusing thing about the loop of Henle is it's this big loop right here, and it goes down deep into the medulla of the kidney. And basically what it does is it creates this high concentration of salt, right? It creates this high concentration of everything down in the medulla of the kidney. So it's pumping out salt and pumping out salt and pumping out salt. And then it reabsorbs it back up here so that things are normal when it gets up here, but there's still a ton of salt down in the medulla of the kidney, if that makes sense. Okay, so it creates a concentration gradient. So up here, everything's not very concentrated. It's like normal. It's like 300 would be normal. And down here, it's like 1200 is the concentration of things. So it's really high down here. And that's basically what you need to know about the loop of Henle, is that it creates a concentration gradient in the medulla of the kidney. Okay, and that's going to be needed later when we get to the distal tubule and collecting duct. Okay, so the distal tubule comes after the proximal tubule, right, after the loop of Henle. And the collecting duct is this big thing down here. These ones are going to reabsorb stuff and do whatever. But the thing about them is that they're controlled. So that means if you're dehydrated, it's going to save water, and it's going to reabsorb a ton of water, and it's going to say, no, we need to keep as much water as we can. We do not want any water to be lost in the urine, right? So it's going to try to save as much water as possible. Your urine's going to be really concentrated, really dark, because it's trying to save as much water as possible. Whereas if you're really hydrated and it doesn't matter, it's going to say, oh no, we can just get rid of all this water. We don't need to work that hard. We'll just get rid of all this water, and your urine's going to be really dilute, really watery, and close to clear. Okay, so the proximal tubule and the loop of Henle, they're just doing their job, and they don't really care how dehydrated or hydrated you are. But the distal tubule and the collecting duct are going to see how hydrated or dehydrated you are, and they're going to decide, okay, no, we need to concentrate urine, right? And if they want to concentrate urine, it's going to makes it possible for them to concentrate urine if they want to, if the concentration down here is really high, right? Because the concentration down here being really high makes it really easy for the urine coming out to also be really high, right? And if we want the urine to be really dilute and whatever, we just plug up all the holes on the membrane so that it can just stay really dilute and it doesn't matter. So they have a choice down here. That's all you need to know. Don't get too confused about it, but they have a choice down here whether they want to be concentrated or not. Okay, so about identifying the tubules, I know everybody gets so confused about this, so hopefully this guide will kind of help you. But so the proximal tubule, the way I identify it is it's a really thick tube, right? And the nuclei are irregularly spattered, so they're not really regular, they're kind of spotted throughout, and it has a brush border. Okay, so this is a proximal tubule. So it is a thick tube, right? This is the middle of the tubule. This is the outside of the tube. It's very thick, right? The nuclei are irregularly spotted throughout, and inside it has a brush border. So you see how it's kind of fuzzy in the middle, and it's almost like it's touching in on itself? That's a brush border, okay? That's a brush border. Whereas the distal tubule looks exactly the same. It's a thick tube with nuclei irregularly spattered throughout, but it does not have a brush border. So this one right here would be a distal tubule. Okay, so the nuclei are irregularly spattered throughout. They're not super regular. It's thick, but there's no brush border. You see how clear the insides of this tube are? That's a distal tubule. This is a distal tubule. You see how the nuclei are irregularly spattered throughout? Irregular. And you can clearly see the inside. That's a distal tubule. Proximal tubules have brush borders. Okay, and this stain you can see right here. This is a proximal tubule. The nuclei are irregularly spattered throughout. It's thick. And you see this darker purple stuff on the inside? That's the brush border. Okay, so if I asked you what this one was, hopefully you would tell me it's a proximal tubule because the nuclei are irregular. It looks just like this one, and it has that darker purple brush border on the inside. Right, whereas this one has nuclei randomly spattered throughout, but it doesn't have that darker purple thing on the inside, so it's a distal tubule. Okay, there's no brush border. right? So this one has a brush border, this one has a brush border. So those are all proximal tubules. This one does not. That's a distal tubule. Okay. When it comes to the loop of Henle, so the ascending and descending loops of Henle, this is the way I remember it. So the ascending loop of Henle, it's going to look bubbly. So do you see how right here, these cells look like they have lots of little bubbles in them? It looks very bubbly. Whereas these big loops right here, it looks very defined. You see how these cells aren't very bubbly? I can clearly trace them. It looks super defined. They look super compressed. That's going to be descending loop of Henle. So I remember descending is defined, ascending is bubbly. So this is ascending loop of Henle. This is descending loop of Henle. Okay. And then, so we already went over distal tubule. So then the collecting tubule, the nuclei are very regular. You see how regular every one of these nuclei are? You see how regular they are? They're in every cell. You can see them evenly spaced throughout, right? They're perfectly spaced in this big circle. So that's how I know it's the collecting tubule. It's very neat, it's very large, and the nuclei are perfectly spaced, right? And then in this section, remember how we said the loop of Henle is down in the medulla where the collecting tubule is going to be, right? So the collecting tubules are all down here. So if I know the collecting tubules are down here, I know that the loops of Henle are going to be down here too. So this is a loop of Henle. Okay, and then there's also capillaries in here. So this is going to be a little capillary. Hopefully that makes sense. This is just to kind of help you guys with this diagram, right? So we've got things constantly being excreted and absorbed and excreted and absorbed. So this is just to tell you in the glomerulus, remember we have filtration. In the proximal tubule, we have active secretion and selective absorption going on. In the loop of Henle, we have passive diffusion going on. And in the distal and collecting duct, it kind of depends. Because again, those are the ones that have a choice. Right, so just look at this diagram and hopefully this kind of helps make sense of what's going on in there. Okay, and then this is talking about that loop of Henle and collecting duct thing I was talking about. So this is the kidney, and this is just showing you the concentration of the kidney in the cortex is around 300. So that's normal. That's what the rest of your body concentration is 300. Whereas in the medulla of the kidney, it's super high. It's up to 1200. And that's because the loop of Henle dips down into the medulla and it creates that concentration gradient. It pumps out all this salt and creates a really high concentration gradient. So that when the loop of Henle goes back up, we've got the distal tubule and then the collecting duct comes down. When the collecting duct comes down, this allows them, if they want, to make a really high concentrated urine. So we can concentrate the urine if we want to. If we don't want to, we just plug up the holes in the collecting duct so that all the salt doesn't rush in and all the water rushes out and stuff like that. But that's a little more complicated than you guys need to know. So it's basically just showing you guys how the thin loop of Henle is dipping down to make that concentration gradient so that when the collecting duct comes down to go outside, it can concentrate the urine if it wants to. If it didn't do that, then it would be really hard to concentrate the urine. Okay, hopefully that helps. And I think that's the end. Yep, that's the end of this review.